Hello, everyone. DNA forensics has revolutionized criminal investigations, solving cases that once seemed impossible. But how did it all begin? And what are the landmark cases that shaped its evolution? Let's dive into the fascinating history of DNA forensics. So, in this lecture, we are going to explore about DNA forensics history and its landmark cases. Timeline 1869 Discovery of DNA. The foundation of DNA fingerprinting was laid in 1869 when Swiss scientist Frederick first discovered DNA, calling it nuclein. 1953 Structure of DNA identified. In 1953, James Watson and Francis Crick uncovered the double helix structure of DNA, explaining how genetic information is stored and inherited. 1970 Discovery of restriction enzymes. The 1970s saw the discovery of restriction enzymes, molecular scissors that cut DNA at specific sites. This paved the way for genetic analysis. 1984 First DNA fingerprinting technique. Alec Jeffreys. A major breakthrough came in 1984 when British geneticist Alec Jeffreys developed DNA fingerprinting, a method to identify individuals using their unique genetic patterns. 1985 to 1990 Use of RFLP. RFP stands for Restriction Fragment Length Polymorphism in DNA profiling. By the mid 1980s, forensic scientists began using RFLP analysis, a technique that compared DNA fragments to differentiate individuals. 1986 PCR Polymerase chain reaction. In 1986, PCR technology revolutionized forensic DNA analysis, allowing scientists to amplify a small amount of DNA for accurate testing, even from degraded samples. 1919 to the present. Development of STR, short tandem repeat analysis. During the 1990s, STR analysis replaced RFLP as the standard for DNA profiling, offering faster, more reliable, and highly sensitive results. DNA forensics. What do you mean by the term DNA forensics? DNA forensics is the use of DNA as an evidence in criminal investigations. It plays a crucial role in identifying suspects, exonerating the innocent, and even solving historical mysteries. Since the 1980s, it has revolutionized forensic science by providing a highly accurate and reliable method for solving crimes. But why is DNA forensics so important? It delivers precise results, making it a trusted tool in legal system worldwide. Beyond criminal investigations, it is also essential in identifying disaster victims and locating missing persons. This powerful technology continues to shape the future of forensic science and justice. History Discovery of DNA fingerprinting. In 1984, Sir Alec Jeffreys, a geneticist, developed DNA fingerprinting, a groundbreaking discovery in forensic science. He found that each individual has a unique DNA pattern, making it possible to distinguish one person from another with remarkable accuracy. This discovery was first applied in criminal investigations and immigration cases, proving its significance in legal matters. Jeffrey's work also led to the development of key forensic techniques. One such method was restriction fragment length polymorphism, or RFLP, which was among the first techniques used for DNA analysis. Later advancements introduced polymerase chain reaction, or PCR, a technique that enabled rapid DNA replication and analysis, making DNA forensics even more efficient. Steps in DNA fingerprinting technique. process. DNA fingerprinting is a multi step process used to analyze and compare genetic material. So let's go through the key steps involved in this technique. Step 1, sample collection. The process begins with collecting a DNA sample from sources such as blood, saliva, hair, follicles, or bodily fluids. Second step involves DNA extraction. Next, the DNA is extracted from the cells using chemical or enzymatic processes. This isolates pure DNA for further analysis. DNA fragmentation. The extracted DNA is then cut into smaller fragments using restriction enzymes. These enzymes recognize specific sequences and create a unique pattern of DNA fragments. Gel electrophoresis. The DNA fragments are then separated based on size using a technique called gel electrophoresis. An electric current moves the fragments through a gel, with smaller pieces traveling farther than larger ones, forming distinct band patterns. Southern blotting and probe hybridization. In this step, the DNA is transferred to a membrane and exposed to a radioactive or fluorescent probes. These probes bind to specific DNA sequences, making the unique patterns visible. The final steps include DNA pattern analysis. The final step involves analyzing the binding pattern of DNA. These patterns are compared between individuals, helping to identify a suspect, confirm biological relationships, or solve historical mysteries. Through this precise and reliable process, DNA fingerprinting has transformed forensic science and criminal investigations worldwide. What do you mean by a term landmark cases? Landmark cases are important legal cases that set new rules, bring big changes to the law, or influence future court decisions. These cases often involve new ideas, new technology, or changing views in society. In DNA forensics, landmark cases have helped shape how DNA evidence is used in solving crimes. They have set important examples for courts, proving that DNA profiling is a powerful tool for finding the truth and delivering justice. These cases have also introduced new forensic methods and legal policies, making DNA testing more reliable. Today, DNA evidence is a key part of criminal investigations worldwide, helping to catch criminals and free innocent people. We are going to explore about two important landmark cases. The first one is the Colin Pitchfork Case 1986 from UK. The second one is the Kirk Bloodsworth Case 1993 from USA. The first landmark case, the Colin Pitchfork Case 1986 UK. In the quiet town of England, two teenage girls were tragically murdered. The police arrested a suspect who later confessed, but something didn't add up. DNA testing was used to compare his genetic profile with the crime scene evidence, and surprisingly, it didn't match. He was innocent. So, how DNA helped? To find the real culprit, investigators launched the first ever mass DNA screening, collecting samples from over 5,000 local men. This groundbreaking approach led to the identification of the true killer, Colin Pitchfork. This case made history as the first criminal conviction using DNA evidence, proving the power of forensic science in solving crimes and ensuring justice. The Cole Birdsworth Case, 1993 USA. In 1984, Kirk Bloodsworth was convicted of the rape and murder of a young girl and sentenced to death. But nearly a decade later, in 1993, DNA testing revealed the truth: he was innocent. Why it is important? This case was a turning point in forensic history. Bloodsworth became the first person on death row in the United States to be exonerated by DNA evidence. His wrongful conviction highlighted the need for a better forensic practices and fair use of DNA in the legal system. As a result, this case led to improved policies for handling DNA evidence in courts, ensuring that science is used to protect the innocent and convict the guilty. Thanks for watching. If you found this video informative, don't forget to like, share, and subscribe for more forensic science content. See you in the next one. Bye bye.